We had just gotten to the point where we were talking about the difference between transcription and translation. So, one of the things that can be challenging is to remember which one is which. Um, Transcription is the name of the process by which messenger RNA is made from the DNA template. That's transcription. Translation is actually another term for building a protein. It is what the ribosomes will do using the RNA as its template in order to make a polymer of amino acids that's known as a protein or a peptide. Okay? So the way I learned the difference between transcription and translation was I just said it over and over again. So I just said transcription, translation, transcription, translation. And I just said it so many times that when I said translation, transcription, it sounded wrong. That's probably not the best way to do it, but that's how I did it. I'll tell you how my TAs tell me that they remember it is when you take something down. If you were to hear someone speak and to write it down word for word, that would be called transcribing someone's speech. Or if I asked you to go into a library and get a book that's written in German, and you're taking a German class right now, then and I ask you, okay, I want you to translate page 236. You might go in there and actually write down page 236, and then you're going to take it home in order to translate the book. Okay, transcription is taking something that is in code, right? Because all of this TACCC, whatever, that is in code, and just kind of transcribing it, it is still in code. Whereas translation is translating the code. The code is in the form of nucleotides, and you're going to translate it into the language of amino acids. So transcription, translation. Now, I was just telling you that ribosomes are going to read 3 nucleotides at a time, and each set of 3 nucleotides in a gene is known as a codon. Right? So here we have got different codons. Each codon is like a little 3-letter code word for one of the 20 amino acids. Right? Let's look at that a little bit more. Oh, yeah. So when we're talking about the genetic code, like you've heard the human genetic code probably mentioned hundreds of times, we're not speaking poetically, we're speaking literally. It took us a long time to figure out the human genetic code. And when we were figuring out the genetic code, we were figuring out, okay, when the DNA says AGG, and when the messenger RNA says UCC, what amino acid does that mean? What is UCC the code word for? Figuring that out was figuring out the genetic code. All right. Now, why do we have a genetic code? Well, let's go back to 2 things. One thing is that proteins are made out of 20 different amino acids. Great. Would it be easy to make copies of things as complicated as proteins directly? Okay, there are a few reasons why no is the answer to that question, but one reason is the strength of nucleotides. Remember, I was just emphasizing that there are only 4 nucleotides in DNA and 4 in RNA. Why? So that those 4 can be as different from each other as black, white, red, yellow. If I told you you were going to have to put 1,000 pieces of paper together in pairs as quickly as you could, as perfectly as you could, and I was giving you your choice of 2 different stacks of paper to do that with, and one stack was going to be black, white, red, yellow, and the other stack was black, white, red, yellow, but also you had to match green with blue, you had to mix aqua with gray, you had to pair orange with peach. You had to pair beige with brown. You had to pair 20 different pieces of paper. You wouldn't even think twice. You know intuitively that black, white, red, yellow, easy to do quickly, hard to make a mistake. Well, nucleotides are black, white, red, yellow. That is why we don't ask our cells to make perfect copies of proteins. But the problem is that if you only have 4 different nucleotides, and that needs to be code for 20 different amino acids, yeah, you're going to need a code. If I asked you to write down this sentence, easy to copy nucleotides with great fidelity, but you can't use letters. You have to use just these 4 symbols. Okay, could you do it? People of my generation actually used to make codes because we played with rocks and sticks for fun. So maybe you've never made a code, but of course you could make a code, right? Now, it wouldn't be, oh, that's A and that's B and that's C and that's D, because, well, you'd be done. Like that'd be the end of your code, right? You couldn't do that. However, you could do it this way. You could do, okay, the letter A, that is going to be triangle, triangle, triangle. And the letter B, I'm going to make that asterisk, asterisk, asterisk. And the letter C, I'm going to make that asterisk, triangle. Oops, I made a star. Asterisk, triangle, circle, right? You could do it. Now, just take it from me that the math shows us that in order to make 20 unique combinations using 4 nucleotides, you need to use them 3 at a time. So we need 3 letter, they're not really 3 letter, 3 nucleotide codons. And 3 nucleotide codons actually allows us to make 64 unique codons. Now, here's something to think about, okay? That by having 64 unique codons at our disposal, how do you think life has managed to organize those codons? It always occurs to me that if this system was being set up by some kind of a committee of humans, and if we had 20 amino acids and we had 64 codons, we would just say simple, we'll just be 3 codons per amino acid, and then we'll have 4 left over, and we'll use the 4 that are left over to say start and stop or something like that. But that is not what happened. And there's a reason why. Even though there are 20 amino acids, and there's a typo here. I'm so sorry. Even though, there we go. Even though there are 64 unique codons and 20 amino acids, it turns out that life has assigned 6 to some of the amino acid. And the reason that that has happened and why that is so kind of brilliant is that these amino acids, when it comes to creating the structure that creates the function for each of these proteins, there are some of these amino acids that are almost impossible to replace. We'll get to there in a second, okay? It turns out 1 is the start codon, 3 are assigned to tell the ribosome, okay, that's it, you're done. And then there are 60 that are assigned to be codons for the 20 different amino acid, but it is not 3 for each one. Oh, I miss this. Okay, at this point in a lecture, I usually say, do we look alike? Because this is my baby. This is actually the protein that I discovered. She's only here because I like seeing her, but also it's a good example of DNA and amino acid sequence. So I'd like you to notice that, like right here, right here, you can see that there are 3 different nucleotides on top of each amino acid. So the letters that are really close together, don't they look like Gattaca? Lots of G's and C's and T's and A's, Gattaca, right? That's the DNA. But you'll notice that under every three nucleotides, there's a single letter. That single letter is the abbreviation for an amino acid. So the DNA looks like Gattaca because it is in code, and that code makes it easy to copy DNA with perfect fidelity, no mistakes, easy to find mistakes when you fix it. And it also will end up making it so that if there is a mistake, and mistakes in DNA are called mutations, that the fact that this is in code and the way the code is organized will minimize the danger of a mistake. All right. So before we talk about that third part, minimizing the effects of mutations, let's finish talking about translation. Transcription creates messenger RNA. And messenger RNA is going to be a complementary little strand that has the instructions for building a protein. That's what the messenger RNA is. This type of RNA, the lowercase r, stands for ribosomal. And ribosomes are made out of ribosomal RNA and protein, because protein does everything. There is a third type of RNA that is necessary for building proteins, and building proteins is translation, right? And that is transfer RNA. Transfer RNA's job is to basically be an escort to a very important amino acid. And I hope I'll be able to show that to you. Okay, so 3 different types of RNA are necessary for the process of translation, which is building proteins. What is not used during the process of translation? DNA is not used. Now, was DNA needed for it? Sure, DNA was needed for transcribing the messenger RNA, but for the process known as translation, DNA is not required. There are other molecules that are related to RNA nucleotides, and they include ATP, which is an energy molecule. NAD and FAD are also energy molecules, and cyclic AMP is a signaling molecule. And these are all related to RNA.